So she says it will go down its concentration gradient. He says it won't. How many agree with her that it will go? There were a few people. How many of you didn't vote? Why not? Yes, sir?
If the question was by facilitated diffusion, then maybe.
But it didn't say simple or facilitated. It just said diffusion, so diffusion could be facilitated diffusion. Yes?
So then yes. So then yes, it would go down to concentration.
Yes, sir?
Does negative and negative repel each other?
Negative and negative do repel each other, which means, so based on the charge you say it won't go inside, they say based on the concentration it will go inside. Yes, ma'am.
It would go inside only if you had active transport because it's diffusion and you went down to the
Why would it go down because of active transport? Because it says the concentration is higher outside. So you're saying just because of the charge it is active transport, but the concentration actually favors it to go in. So why do we need active transport? Aha! But you don't know if they need active transport. So you are correct that you need to pay attention to both the electrical gradient and the concentration gradient. If you were looking at charge alone, you would say the charges would favor it to move out, therefore you need active transport. But charge is not everything, it's also the concentration. And the concentration in this case favors it to move in. So if you look at your diagram again, the concentration favors the movement in, the charge gradient, the electrical gradient favors it to move out. So which direction is it going to go?
Neither.
There is not enough information for you to figure that out. So the answer is, it depends. What does it depend on? Yes, ma'am. No, ma'am.
Ma'am.
So it absolutely does depend on the electric chemical gradient, which means you have to know which one is stronger. You have to know whether the electrical gradient is stronger or the chemical gradient is stronger to know which direction it would be moving down diffusion. And then you would know if it's going to, diffusion favors it to move out, then inward movement would be active. If the diffusion favors it to move in, then inward movement would be passive. But you need to know which one is stronger. And I didn't give you enough information to know that. Yes, sir.
Could we say that the pressure potential has an effect? Could we say that pressure potential on the outside of the cell has an effect on whether or not the ions go inside or outside, or
The pressure potential?
yes, maybe they're forced through some kind of force inside the cell, or the size of the ion itself.
In plant cells, pressure potential does matter because of the way that the turbo pressure works to push the vacuum against the cell wall. And I have not told you anything about that. So I'm impressed that you even brought up pressure potential, but you're not going to do anything with that till Bio 460. 460, sorry. You have a huge presence. But you will actually take a look at how you figure out which direction nitrate is going to move in Bio 207. There is a special equation called the Nernst equation, which we are not going to use in this class, and I'm not going to tell you how to do it. Where you will look at the concentrations of different ions across the membrane and calculate based on that which direction your ion of interest is going to move. And you'll do that in Bio 207. You're not going to do it in Bio 101 or Bio 102. So yeah, how about why? But if I gave you an exam question like this one, this were an exam question, your answer would be,
It depends.
and then you would explain what it depends on. What does it depend on?
Depends on it, whether the chemical or the electric is.
It depends on whether the chemical gradient is stronger or the electrical gradient is stronger, and that will tell you which direction diffusion would favor the movement. Then you would be able to figure out whether movement into the cell would be active or passive. So remember, for ions, you have to look at both the concentration gradient and the electrical gradient. You can't make a decision based on one alone. So far, so good? All right. Now, sometimes you have a protein that's moving two things at the same time. You saw one example with the sodium potassium pump. In some cases, you are getting the active movement of one coupled to the passive movement of another one. In the sodium potassium pump, they were both being moved actively. But a more common example is to move something actively and couple the energy requirement of that to the movement of something else that is moving passively. So the passive movement releases energy, the active movement requires energy. So a typical example is what plants and bacteria and fungi do, which is they pump protons across the membrane, so that's active transport. And they allow the protons to go back down their gradient and couple that to movement of something else against its gradient. So let me show you an example. So if we take a look at this diagram here, here's the proton pump. The proton pump uses ATP hydrolysis, and that provides the energy to move protons against their gradient. Typically, much higher concentrations of protons outside of the plant cell than inside. So the cell walls of the plant cells tend to be quite acidic compared to the inside of the plant cell. And then there are these co-transport proteins that allow the protons to move down their gradient back into the cell. That releases energy. And they use that energy to bring something else against its gradient. So sugar movement, for example, into plant cells is coupled to the movement of protons. Protons are moving down their gradient, sucrose is moving against its gradient. The movement of protons releases energy, the movement of sucrose requires energy, couple the two of them together. You don't need to have a special sucrose pump or a special, let's see, I think there's a nitrate pump. There are a whole bunch of co-transporters in plants that use the movement of protons that they only have one pump, and then you just have modified versions of these transport proteins that bring other stuff in, as opposed to having 80 different pump types. So this diagram, which is from a plant physiology textbook, is just to show you how complicated transport into and out of a cell can be. So we just did a very, very brief, simple overview of that. It's showing you all of the different types of co-transporters, which are moving things at the same time in the same direction or in opposite directions. So you can see lots of them have protons coupled to something else. So you have protons sucrose, proton amino acids, protons peptides, protons and sodium. In this case, they're going in opposite directions. Protons and phosphate, protons and nitrate. You have proton pumps here, and then the vacuole has its own set of transport proteins. So things get to be quite complicated. And so if you go back and think, all right, we're going to use that equation that I mentioned and try to figure out if something is going to move down its gradient or against its gradient, you have to know all of these different ions that are going into or out of the cell, their concentrations inside or outside of the cell, and then figure out for your ion of interest which direction is it going to be. So things can get very complicated. So I don't want you to come away thinking, oh, I know everything there is to know about transport across the membrane. We just did this much out of something the size of this campus. So far, so good?
Yeah.
Yes, ma'am. They're all over the place. This diagram is showing you one example of each. They're very neatly organized in here just to show you the different types of transporters you have, but no. And they're not all present all the time, so the plants can actually synthesize more of the different transporters that they need at different times and insert them into their membranes and then remove them. Same thing with aquaporins. They can add more aquaporins when they want to move more water, and they can remove them when they don't want to. They regulate which transporters are present at which times. And not all cells use them all at the same time? Good questions. All right, so we are going to switch gears and move on to our introduction to metabolism and enzymatic reactions. So I did post a list of topics you should focus on as you read the modules before the exam. I'm sure nobody did, given that you had an exam on Monday. So you'll be doing some catching up this week. And depending on how much high school chemistry you have, some of the basic stuff that you read in the modules could be reviewed. Hopefully you know what the word entropy means. All right? Yes, define entropy for me. All right, so disorder in the universe is called entropy. So when you deal with spontaneous processes that go forward without any additional input of energy, that means that you're carrying out a process that will increase the entropy of the universe. So the disorder in the universe is increasing. And we're going to talk about spontaneous reactions. And typically, spontaneous reactions, we're talking about catabolic as opposed to anabolic reactions. So you should know what catabolic and anabolic mean as well. You hopefully have heard of free energy and Gibbs free energy. Yes? Some of you, maybe. So when we talk about free energy in terms of reactions, we always look at the change in the free energy of a system. So the system being your cell, and whether the free energy, which is the capacity to do work, is increasing or decreasing, depending on what type of a reaction you're carrying out. So remember this from high school chemistry? All right. Be relieved, I will not ask you to calculate delta G. You do have to look at diagrams that show you negative or positive delta G values, but you don't have to actually calculate it in this class. But when you take chemistry, you do have to do that. So Gibbs free energy, which we are only interested in the change in Gibbs free energy in a system as reactions go forward or in opposite direction. So we're looking at the total energy change and the change in the entropy, which is the increase or decrease in the disorder, which is coupled with absolute temperature here, with T in Kelvin. If the delta G comes out to be negative, that means the process is spontaneous. If it turns out to be positive, it's non-spontaneous. So don't worry about calculating this. We will not ask you to calculate that. But if you are curious, go back to your basic chemistry textbook and review what Gibbs free energy is. What we need you to know is that when we talk about reactions, we always talk about whether this reaction is spontaneous or not. If it is spontaneous, that means you have a negative change in free energy. So you started out with more free energy and you end up with less free energy, so you're releasing energy to the environment. If you're looking at a non-spontaneous reaction, that means it takes free energy from the environment because the free energy of the products are higher than the free energy of the reactants. That's about as much as we need you to know in terms of Gibbs free energy. So more similar. All right. You do need to know these two terms, exergonic and endergonic. Exergonic reactions are spontaneous because they release free energy. They have a negative delta G. Endergonic reactions are non-spontaneous because they require energy, because the delta G is positive. So the reactants, in the case of exergonic reactions, have more free energy than the products. In the case of the endergonic reactions, the products have more free energy than the reactants. So graphically, we're going to take a look at what that looks like. But before we do that, you also have to know another term. Activation energy. Any reaction, regardless of whether it's spontaneous or non-spontaneous, requires an initial input of energy, and that's called the activation energy. So that your substrates reach a transitional state at which the chemical bonds can be broken and reformed. So let's take a look at this diagram here so that you can see what I'm talking about. On the y-axis,